Hello everyone, in this video we're going to do a review of Mendelian genetics. My first suggestion for you is if you weren't very comfortable with mitosis and meiosis, and if you got a lot of questions wrong when we did the test on those material, you should watch some of these videos to help yourself remember what mitosis and meiosis is about. But otherwise, let's get started. The first question is the difference between mitosis and meiosis. I attached a picture over here to help you remember what it's about. So we start, let's say, a parent somatic cell. Remember, somatic cell is a body cell, so that has to be 2N and diploid. If you were to go through mitosis, you're making a copy of whatever cell that you start with. So if you start with a 2N cell, you still end up with a 2N cell. You make an exact copy of the parent cell. And then for mitosis, there are 4 steps. We have the punnett, right? Prophase, metaphase, anaphase, and telophase, and the last one is cytokinesis, where the 2 cells actually separate. But for meiosis, we do this twice. We do pmat1, and then we do pmat2. So the cell separates twice. So let's first take a look at mitosis. We start with a 2N cell. What happens here is that the sister chromatids, remember the sister chromatids, are the 2 exact copies of the DNA attached at the same point. So it's an X shape. These 2 are sister chromatids to each other. So the sister chromatids line up at the metaphase plate during metaphase. During anaphase, the sister chromatids separate, and then telophase, it keeps on separating, and cytokinesis actually separate the 2 cells into 2 cells. So now we have 2 exact copies of the original parent cell. But for meiosis, instead of having sister chromatids lining up, during metaphase 1, we have homologous chromosomes. So those are chromosomes that can pair up with each other. Homologous chromosomes line up at the metaphase plate. So if you look at this one and this one right here, it looks different, right? And then during anaphase, the homologous chromosomes separate, and then telophase, it keeps on separating. The result is we start with 2N, but by the time we get to here, by the time we get to the end of meiosis 1, we only have 1M. Now we have a haploid cell. Now you can take a look at the meiosis 2. So meiosis 2 is pretty similar to mitosis, where you have sister chromatids lining up at the metaphase plate during metaphase 2. And the anaphase, the sister chromatids separate, and then the result is we get 4 cells at the end of meiosis, and the 4 cells are all different from each other, and they're all haploid cells. They only have 1 copy of each chromosome. Right here we have 2 copies of each type of chromosome. Okay, 2N to 2N, or if we start with 1N for mitosis, we end up with 1N as well. But for meiosis, you always have to start with 2N, and then you end up with 1N. So I did this pretty quickly. If you're not really sure what was going on, make sure you watch these videos so that you can review this part a little more slowly. But if you are good with what's going on here, we can continue on. So here there's some differences between mitosis and meiosis. What are they? First off, what do we start with for the parent cell? For parent cell, mitosis can start with haploid or diploid. Meiosis has to start with diploid, right? And then the daughter cells produced for mitosis, if we start with a haploid, we end with a haploid. If we start with a diploid, we end with a diploid. For meiosis, we start with a diploid and we end with a haploid. Chromosome number is different or the same to parent cell. For mitosis, it's the same. For meiosis, it's half of the parent cell. Okay. Make sure you understand, if you are able to draw this entire thing by yourself, which you should, then you should be good. So here are some other things that you want to make sure that you really understand. For a chromosome, one chromosome can either look like this or look like this. So this is an unduplicated chromosome. If you have an X shape, that's a duplicated chromosome. So this is one chromosome. This is still one chromosome. But right here, we don't have any sister chromatids. But since we have an X shape, we have duplicate of the same DNA, then we have 2 sister chromatids right here. All right, the next one is what happens during interphase of the cell cycle. If you remember, the cell cycle has 4 steps, G1, S, G2, and M. So G1 is a gap 1 phase where the cell grows and getting ready for mitosis. And then S phase is where DNA replicates, so it goes from here to here. And then G2, we're still preparing, and then the M phase is the mitotic phase where we do the PMATC, okay? So the interphase, the cell grows, DNA duplicates, organelles duplicate. What happens during the mitotic phase of the cell cycle? Mitosis. What is apoptosis? So apoptosis is a programmed cell death. That's where the cell, with the gene is programmed to kill that specific cell. Why is it important for organisms? So for example, when our fingers are growing, during embryonic development, it starts with a webbed finger. So our fingers are like duck fingers. They're all webbed together. And apoptosis allows the formation of our actual fingers. The webs in between the fingers disappear. The next one is, how are the growth of a tumor and repair of a scrape on the knee similar? They're both replications of cells, right? If you scrape your knee, then you have to make new cells to make up that part of the scrape. How are they different? A tumor or a cancer are unregulated cell duplication. So the cells keep on replicating and it'll never stop. That's a tumor or a cancer. But repair of a scrape on your knee is regulated cell cycle, regulated cell duplication. So once your knee is fine, it will no longer be making new cells. So that's the difference. Next one is cell differentiation. Cell differentiation starts with something called a stem cell. A stem cell is an undifferentiated, unspecialized cell, meaning that it doesn't really have a specific function. It's not doing anything yet. And it has plenty of potentials to develop into other things. And cell differentiation is what allows the stem cell to turn into all the different kinds of cells in your body. So you would have stem cell in your spine, adult stem cell, but then they can turn into certain types of blood cells. But if you have embryonic stem cell, the stem cell before a baby is born, then it can turn into every single type of cell in your body. It can turn into eye cell, lung cell, whatever. All right. The next one is, what is the number of chromosomes in a normal human body, or we call it somatic cell? So you need to know body cell is also the same thing as somatic cell. The number of chromosomes in a normal human body cell is 23 pairs. So if you look at this one right here, we have 22 pairs, and then we have the X and the Y. So it's 23 pairs, or you can call it 46 pairs of chromosomes. So what do we call this one and this one? If they can pair up, we call it homologous chromosome, right? If we have the X shape, each part of the X is called a sister chromatid. All right, a human body cell or a somatic cell, is it haploid or diploid? Remember, haploid means you have one set of chromosomes. Diploid means you have two sets. So right here, we have two sets, right? For each one of them, we have one set and then two sets of chromosomes. So we call it a diploid. What is the number of chromosomes in a normal human gamete? A gamete is either a sperm or an egg. So if you think about it, a sperm and an egg needs to have half of the amount of your normal body cells, so that when a sperm and egg combines, you can get the whole number, the 23 pairs. So for each gamete, we have 23 chromosomes. Not 23 pairs now, we only have 23. So if you look right here, we have 23, and the last one could either be an X or a Y for our for a sperm. But if we have an egg, it has to be an X. Be able to count the number of chromosomes when you're given a picture of a cell. If you see something like this, that's 2. So this is 26 chromosomes. But if we have right here, that's 1 chromosome. This is also 1 chromosome. So you need to be able to count the number of chromosomes. The next one, what are chromosomes? They are DNA wrapped around proteins, and they code for something, or it might not. But for the most part, in our mind, the DNA or chromosomes code for something that has a function. How does the structure of a chromosome differ in prokaryotes and eukaryotes? For prokaryote, we usually have a circular DNA, circular chromosome, and for eukaryotes, we have linear. Next one, be able to identify all these words. So let's take a look. A centrosome, during mitosis and meiosis, a centrosome looks like this. It's kind of a little T shape. And what they do is that they shoot out these spindle fibers, because those spindle fibers can attach to either the sister chromatins or homologous chromosomes, and then separate them during anaphase. See over here, the spindle fibers pull the sister chromatins in this picture apart. And the nuclear membrane is the membrane of the nucleus. A homologous chromosome, we already talked about that. Sister chromatins, we talked about that. So we're good. How do you tell if something is mitosis, is going through mitosis or meiosis? Usually if we have sister chromatids aligned in the middle, that's either meiosis 2 or is mitosis. But if you see homologous chromosomes lined up, like right here, homologous chromosomes, those pairs of chromosomes line up in the metaphase plate, then that has to be meiosis. And the next one, draw each phase of mitosis and meiosis. I already told you, you have to do that. What happens during each of the 4 phases of mitosis? We already talked about that as well. But if you're confused about the difference between mitosis and meiosis and how you can identify them, you can take a look at this part, which I'll post in our shared answer key as well. What is cytokinesis and when does it occur? So cytokinesis is when the two daughter cells actually separate, completely separate. When does it occur? It occurs after anaphase and telophase. And it happens in both mitosis and meiosis. What would happen if the spindle fibers, right? The spindle fibers are these little pulleys. What happens if they are disrupted during metaphase? What happens if they don't separate the sister chromatids or homologous chromosomes correctly? Then you'll get something called non-disjunction. Non-disjunction will result in a wrong number of chromosomes within the daughter cell. So for example, right here we start with 2 chromosomes, and in the end, in all of our daughter cells, we should end up with 2 chromosomes. Yeah, anyway, but the result is right here, we have a not normal amount of chromosomes. That's what it is. We have the phases of meiosis, PMAT C. So prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. Prophase 2, blah, blah, blah. The next one, during which phase of mitosis and meiosis are chromosomes lined up at the metaphase plate? Or you can say lined up at the middle of the cell. Then it has to be metaphase. And then the last one is what actually lines up for mitosis and meiosis. Mitosis, always, always, always sister chromatids. Meiosis, if it's meiosis 1, then it will be homologous chromosomes. If it's meiosis 2, then it's sister chromatids. What type of cells are produced by mitosis? It can be haploid or diploid as long as it's the same exact cell as the parent cell. Meiosis, you always have a haploid daughter cell. You get actually 4 daughter cells for each parent cell. Difference between, so we pretty much did this. Haploid and diploid. Haploid is 1 set of chromosomes. Diploid is 2 sets of chromosomes. So if a haploid cell has 20 chromosomes, and then the diploid cell has to have 40 chromosomes. Or if I tell you a chicken has, I don't know, I'm just making up a number, 36 chromosomes in their somatic cell or body cell, then what is the amount of cells produced in a chicken gamete? And then the chicken gamete has to have half of that, so 18, and it's haploid. How about a baby chicken? Then it has to be also 36. For mitosis, we did this. For mitosis, the parent cell can be haploid or diploid. The daughter cells can be haploid or diploid as long as it's the same as the parent cell. For meiosis, parent cell has to be diploid, daughter cell has to be haploid. What is the difference between a somatic cell and a gamete? Somatic cell is your body cell, and it has to be diploid. A gamete is your sperm or egg, and it has to be haploid. Mitosis produces what type of cell? Mitosis produces your body cells or somatic cells. Meiosis produces what type of cell? Then that's your gametes, because your gametes are haploid. Meiosis produce haploid cells. And the next one, what gets separated during anaphase of mitosis? Back to this again. As long as you remember this picture, you can answer all of these questions. If you can draw this out, you can answer all of those questions. Going through it once again. During anaphase of mitosis, sister chromatids get separated. During meiosis 1, homologous chromosomes. How about meiosis 2, sister chromatids? All right, now finally something new. What is crossing over? Crossing over happens only during meiosis, because only during meiosis do we have homologous chromosomes pairing up. But when homologous chromosomes are paired up, they're so close to each other that a lot of the times a part of each homologous chromosomes will do a crossing over just like this. It switches apart. So these two are homologous chromosomes. These two are sister chromatids. These two are sister chromatids as well. But these two are non-sister chromatids. Those get switched. And what phase does it happen? It happens during metaphase 1. What is independent assortment and how do they contribute to genetic diversity? It increases genetic diversity, but how exactly does that work? During meiosis, you can have a cell like this, right? But for a human, we would have 23 pairs. But for each one of the pair, you can either line up this way, if we were to only have 2 pairs. You can either line it up this way during metaphase 1, or you can line it up like this during metaphase 1. So the result is we can either get those daughter cells, or we can get those daughter cells. So independent assortment means the chromosomes, the non-homologous chromosomes. Wait, the homologous chromosomes are in, no. The non-homologous chromosomes, so these and these are non-homologous chromosomes, right? They are independently assorted into the daughter cells. So you can either have these or this. All right, you can have different types of lineups. So make sure you understand that. The next one, compare mitosis, meiosis. Which one allows more genetic diversity? Of course, it's meiosis because of all the independent assortment and crossing over and fertilization, which hasn't happened yet if it's only meiosis. Those allow more genetic diversity. If you have mitosis, then you're only making copies of the cell. Then you can only have genetic diversity if there's a mutation, which, I mean, it happens a lot, but it's not as much genetic diversity as meiosis, not nearly as much. But having those genetic diversity is always good. Being able to respond to the environment when the environment changes is always good, because if everybody is the same, something happens, everybody dies. But if it happens that there's some individuals that are more fit for the changed environment, then that's good. Some will be able to live. What do prokaryotes go through in order to allow cell division? That's called binary fission. All right, here's another picture showing you meiosis and mitosis. Mitosis only separates the cells once, cells divide once, and meiosis, cells divide twice. Make sure you can draw this. Again, make sure you can draw this. Draw it. All right, we have a few more left. What plant did Mendel use to study genetics? Is the pea plant. Now we have a bunch of vocabularies, okay? You need to make sure that you understand these vocabularies so when you see those words in a question, you know exactly what it means. So the first one is, what is a gene? It's a sequence of DNA that codes for a protein or a trait. For example, you can say an eye color gene, that is a sequence of DNA within your body, within a cell, that codes for eye color. We can have hair color gene, we can have blood type gene, all of that. How big your nose is gene, but I'm sure that's not just one gene. So that codes for a trait or a protein. What is a genome? Those are all of the genes in a cell or organism. So when we talk about a human genome, that's all of the genes within your body. What is a locus? Let's first take a look at this one right here. For a flower, we can have the flower color, right? And we can have the flower color gene. So the locus is the location of a gene. Where is it located? Within on a chromosome. So we can have a flower color gene right here. We could potentially have a leaf size gene right here. So that's where it is on the chromosome. Next one is the allele. Allele is a version of a gene. So for eye color, for example, you can have the brown eye allele, you can have the blue eye allele, you can have that. Let's say brown eye will be big A, blue eye will be little a. So that's the A's, the letters. So alleles, we can have an allele for white flower and an allele for purple flower. For a dominant, allele will be like a big A. And that would, if you have big A, then you still have brown eye. And big A, little a, that's still brown eye because brown is the dominant allele. Recessive, you only have blue if you have two recessive alleles, little a. So allele is one version of the gene. This is one version, the brown eye allele. This is another version, the blue eye allele. Make sure you are very clear with these words. Homozygous is when you have two of the same thing, so big A, or little a. Heterozygous is when you have two different alleles, big A, little a. You have homozygous dominant, and you can have homozygous recessive. Genotype are genes or letters, so big A will be a genotype. A phenotype is the actual trait that you can see. So for example, the brown eye is a phenotype. A monohybrid cross is when you are only looking at one trait, so just the eye color, not eye color and hair color. That would be a dihybrid. You can have P generation, that's the parental generation. So let's say you have these two as the parents, that's the parental generation. F1 generation, if those two cross and they have babies, and all the babies are going to be big A, little a, those are going to be the F1 generation, the first generation. And the F2 generation is the second generation after that, so the grandkids generation. Purebred means the same thing as homozygous. It could be either big A, or little a, dominant or recessive. Autosome and sex chromosome. So autosomes, if you look right here, autosomes are the ones that are not determining your sex. So for a human, from 1 to 22, all those are autosomes. Sex chromosomes are the XY and the XX. So for a male, it's XY. For a female, it's XX. It determines your sex. And then sex chromosomes. All right, the next one, how many alleles of one gene, let's say eye color gene, does the diploid parent cell have? Diploid means you get two copies of the same genes, two sets of chromosomes. So diploid parent is going to be big A, for example, and you get two alleles of one gene. This is one allele. This is one allele. So one letter basically is one allele. How about a gamete? A gamete has to be a haploid cell. So now you only get one of those. So this is one allele of one gene. But if you have a parent that's big A, little a, then this could be big A or little a. Allele for the gamete, right? So this is why you have to understand mitosis and meiosis, because in order to do Punnett square, understand how Punnett square works, you have to, those two are related. Then the next one, an offspring inherits one allele from each parent. So if you look at this picture right here, we have mother, big B, little b, father, big B, little b. For each kid, because the gamete only carries one gene, well, one allele of the same gene, right? You either get big B for the gamete from mother, the egg can either have big B or little b, and the sperm can either have big B or little b. And for a kid, big B, little b, for example, this kid right here, he or she gets one allele from mother, one allele from father. So two haploids together, and then we get one diploid for the child. The next one, independent assortment, we already talked about that. What factors determine genotypes? What factors determine phenotypes? Remember, genotypes are only the genes. The letters determine the genotype. But phenotype are genes and the environment. So for example, how tall you are is not just your genes, it's your genes and the environment. But there are also other factors, such as your eye color will probably stay the same your entire life, no matter what the environment is, unless something bad happens. What does it mean when something is sex-linked? When a gene is sex-linked, that gene is located on the X chromosome. So instead of being on one of the autosomes, the 1 to 22 pairs, it's on the X chromosomes. So for a female, you get 2 X's, and you get 2 copies of the same gene, 2 alleles. But for a male, since you have X and Y, you only get 1 allele. Make sure you can do a Punnett square. So you need to know X is a female, XY is a male. I shouldn't have to tell you that in a question, and I will not tell you that. So you know that. All right. An example given to you over here is X big HY and X big H and X little h. Which one is the mother? Which one is the father? Think about it. This one has to be the mother. This one has to be the father. So if I say the mother has a sex-linked trait and the mother is heterozygous, then you'll know it's X big H, X little h. But if the mother is homozygous for the trait, then it's X big H. You write it as a superscript and then X big H. In this case, the father is also X big H and Y, and you get these 4 kids, potential kids. What is the probability that you can have? So I can ask you all kinds of questions, right? What is the probability that the child is a male? It's 50%. What is the probability that the child has the dominant trait? You get 1, 2, 3. That's 75%. What is the probability that the child is recessive, has the recessive trait? Then it's 25%, right? So you can do all kinds of those. So here I have a link to a website with more Punnett square questions right here. So you can do some of these, and we had some worksheets that we did in class that were really good that you can do some practice, because there's definitely going to be a Punnett square question on the test. But here I have an example. You can keep watching, and I would suggest you to pause the video and try to do it on your own, and then come back and check your answer. So let's talk about this. One cat carries heterozygous long hair trait, big S, little s. So even if I don't tell you this is big S, little s, if I say the long hair trait is dominant over the short hair, and one cat is heterozygous, you will know that it's big S, little s, right? So now one cat is big S, little s. And then this mate carries homozygous short hair traits, even if I don't tell you little s, but if I tell you long hair is dominant, you will know that the mate is little s, so we'll do little s, little. Okay. Use a Punnett square to determine the probability of one of their offspring having long hair. So now we set up the Punnett square, now we can make it better. All right. Having long hair, so you know that these 2 are long hairs. So the probability would be 50%. All right. Make sure that you do some of these questions as a practice, either tonight or before our test, to make sure that you know how to do these type of questions.